Good morning. It's July 24th, and this is your daily brief in science. Here's everything you need to know. A historic moment in mathematics as Hong Wang and Yu Deng of Peking University, alongside two collaborators, win the Fields Medal, the first time Chinese nationals under 40 receive mathematics' top prize. Their work on Hilbert's sixth problem builds a rigorous bridge between different scales of dynamics and offers a unifying framework across mathematical physics. The announcement came at the International Congress of Mathematicians in Philadelphia, marking a milestone in unifying diverse mathematical and physical theories. The story situates these laureates within a broader regional ecosystem that ties Guangdong's development model to talent cultivation and a growing AI and tech landscape. The spotlight on AI discusses two roles. AI can suggest proof paths or conjectures, while humans handle verification, explainability, and assessment of significance. The profiles emphasize long-term dedication, collaboration, independent problem-solving, and passion beyond rote study in scholarly work. This reflects a coordinated national effort to nurture talent, expand top universities, and balance foundational research with applied tech growth. Zimmerman's move to OpenAI's safety division signals how AI tools are reshaping research careers, including in mathematics. The context underscores the ICM's focus on scientific progress, with AI as a major theme and notes of AI-driven progress on classic three-dimensional problems. China's innovation capacity is highlighted through robust infrastructure and an integrated ecosystem that supports breakthroughs in emerging tech and fundamental science, with reporter Li Su-min providing context and profiles. Deng and Wang's work connects geometry and physics, touching on symplectic geometry and potential links to string theory. A new study finds that GLP-1 receptor agonists may carry a higher risk of hair shedding than some other diabetes medicines, though the overall risk remains small. The research compares hair loss rates per 1,000 person-years and shows about 7 for GLP-1 agonists versus 5 for SGLT2 inhibitors, and roughly 7 versus 3.9 for GLP-1 agonists compared with DPP-4 inhibitors. Most cases are described as temporary non-scarring shedding, with weight loss, nutritional issues, or hormonal changes suggested as possible mechanisms. The exact cause is not clear, and authors call for more research to understand biological factors and risk modifiers. The study aligns with FDA observations and emphasizes that clinicians and patients should consider this potential side effect in shared decision-making. Importantly, the article notes the absolute risk is small and does not provide specific management guidance beyond informing patients about the possibility. Details such as sample sizes or precise statistics are not included in the excerpt. Background on author Alice Barraclough is mentioned, but the study findings remain the focus. Related separate research hints at possible reductions in hospital admissions for alcohol use with these medications, though evidence is limited and inconclusive. Findings sit within ongoing regulatory inquiries and prior case reports, underscoring the need for more research on mechanisms and risk factors. Researchers emphasize that rapid weight loss and nutritional issues could explain the temporary shedding, but it remains unclear whether the effect is reversible after stopping or continuing therapy. Nuvectis has announced that ciprocopon, also known as NXP100, received marketing approval from China's NMPA to treat paroxysmal nocturnal hemoglobinuria in patients who have not previously used complement inhibitors. The approval follows a Phase 3 trial in treatment. Naive PNH patients comparing ciprocopan directly to eculizumab, with ciprocopan meeting all primary and secondary endpoints. In the head-to-head study, 59.5% of patients reached a 12 gram per deciliter hemoglobin target on ciprocopan versus 8.3% on eculizumab, and baseline hemoglobin rose by about 5.0 grams per deciliter versus 2.2 grams per deciliter with eculizumab, along with reduced transfusion needs, 94.6% not requiring transfusions versus 69.4%. The trial also showed superiority over Soliris in the primary and secondary endpoints, supporting the approval based on these direct comparisons. Nuvectis independently developed ciprocopan and will manage development, regulatory approvals, and commercialization in China. while Hisco developed the drug and retains regulatory and commercialization responsibilities within China under a June 2026 license that grants Nuvectis exclusive rights outside Greater China, India, and parts of Southeast Asia. Nuvectis holds exclusive development and commercialization rights outside those regions, with Hisco continuing regulatory work and commercialization in China. The broader pipeline includes NXP 100, ciprocapan, along with NXP 900 and NXP 200, all focusing on complement-related conditions and oncology, reflecting Nuvectis's ongoing development strategy in this area. Management and Hisco executives expressed optimism about ciprocapan gaining market share in the evolving Factor B inhibitor landscape and expanding beyond PNH, while the press release notes standard forward-looking statements and safe harbor language. A new study uses single-cell multiomics and AI to connect how the genome's three-dimensional folding relates to gene regulation in Alzheimer's disease, offering a multi-scale view of disease progression. Researchers applied gauge-seq to measure gene expression and 3D chromatin contacts in the same cell, and integrated chromatin accessibility and spatial transcriptomics in intact prefrontal cortex tissue. Across major brain cell types in AD, they observed a reproducible shift in genome contact patterns, reduced short-range and increased long-range interactions, with weakened compartment segregation as active and inactive regions mingle more. The work links 3D genome remodeling to cell type-specific transcription and tissue architecture, establishing genome folding as a regulatory layer in AD pathology, and a framework for identifying regulatory elements in gene programs for future studies and therapeutic exploration. The study was conducted by teams from Carnegie Mellon University, the University of Pittsburgh, and the University of Washington and published in Science with NIH funding. An AI model named HickFormer combines DNA sequence, genome folding features, and local 3D contact maps to predict cell-type-specific gene activity and test how structural changes drive transcriptional programs. Predictive modeling shows 3D genome features add explanatory power beyond sequence alone for AD-related gene expression, enabling prioritization of distal regulatory elements mediated through chromatin contacts. A key finding is increased compartment mingling, where A and B compartments lose clear spatial separation, correlating with reduced overall gene expression and disrupted cellular function. At promoter proximal regulatory elements, interactions weaken, while mid-range regulatory interactions become more prominent at sites tied to chromatin loop organization. Senior author Jian Ma emphasizes integrating genome folding with cell state and tissue context to move beyond cataloging changes toward understanding mechanisms and informing next steps in research and therapy. The study's map links 3D genome remodeling to tissue architecture in AD, providing a platform to identify regulatory elements in gene programs for functional studies and potential therapies. A large clinical trial over years tested whether Prosigna-guided treatment could spare chemotherapy in high-risk early-stage ER-positive, HER2-negative breast cancer. The Optima study enrolled 4,429 patients across multiple countries and followed them for a median of 4 years, providing Level 1A evidence for the test's ability to predict chemotherapy benefit and support safe de-escalation of treatment. Prosigna calculates a risk of recurrence score from tumor analysis, using machine learning to estimate long-term outcomes and recurrence risk, guiding decisions for this breast cancer subtype. Developed with researchers including UNC geneticist Charles Perrault and marketed by Veracyte, the test is used by clinicians in the United States and United Kingdom to tailor therapy and reduce unnecessary chemotherapy while preserving outcomes. Optima found that 68% of clinically high-risk patients could safely forego chemotherapy, with a 5-year cancer-free survival rate of 93.7% when treated with hormone therapy alone. The results showed efficacy across diverse groups, including premenopausal women and patients with lymph node involvement, indicating broad applicability. These findings were presented at the 2026 ASCO annual meeting in Chicago on May 31st. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.